So I'm really delighted to be here to address this audience. A little bit intimidating being the first speaker of this series, but I'll do my best to keep your attention. My approach to, over my entire career, to trying to answer complicated and tough questions about mechanism, whether that be a physical mechanism in nuclear medicine of disease progression, or whether in genomics, looking at mechanisms of transcriptional behaviors and their implication on health, disease, and so forth, is I'm interested in the limitations of the measurements. So how can we extract a signal from a very noisy measurement, whether that be a measurement of a transcriptional nature or a chromatin conformation nature. The properties of the measurement are what are going to determine the ability to be able to say something about mechanism. And so the mathematics comes in looking at two things. First, fundamental limits, phase transitions. When do you have enough information, enough data, a high enough signal-to-noise ratio to be able to reliably extract information from the data? And then how do you act on those phase transitions to produce algorithms that can extract the information in a reliable manner? So that's going to be sort of the theme here. That's the word inference, right? Inference means inferring from data information that's relevant to your objective of understanding mechanism. Multi-layer and multimodal refer to the tremendous explosion of multiple views in the case of genomics through the wonderful technologies that we have, which all measure different aspects of transcriptional, metabolic, and other factors. So I'm going to go through this from a fairly high level, starting with sort of a general review of multi-layer networks. Then I'm going to talk about community detection as a way of extracting coherent submodules of networks that interact together from the background of perhaps non-specific spurious interactions that constitute communications between communities. So they could be intentional but spurious in the sense that the community itself is very strongly and tightly coupled, right? The nodes in the network are very tightly connected. And then I'll talk about these phase transitions that will determine what are those fundamental conditions that we need in order to be able to reliably extract the existence of a community from a large network, and then identify the nodes in that community. And then I'm going to talk a little bit about multimodal relevance networks, which is acting on a probabilistic model for the multi-layer multimodal network with specific application to integration and aggregation of form and function types of measurements from Hi-C and RNA-seq. So, multi-layer multimodal networks. This is a cartoon that shows what I'm talking about. There's several ways that multi-layer networks can arise. If you look at a simple network with a fixed number of nodes, and its edges, which correspond to interactions, dependencies, communication, between these nodes, which could be genes, it could be agents in a general network, a social network, or an engineered network like a communications network with the internet, where these would be IP addresses and communications between them. And over time, the dependency relationships, the communication patterns change. So these are multiple layers. Each time slice is a particular layer. The nodes, therefore, are multiplexed throughout the layers with edges that change over time. And this can be contrasted with another form of multi-layer networks, which are multimodal, in that the edges may have different meanings. Right here, the edges might just be communication, right, between these nodes over time. But here, one edge might be one type of communication, and another edge might be another. So in a social network, these might be individuals who are friends on the red lines, and they're related through a blood relation on the green line. So those are two different fundamental relationships. Handling the extraction of community when you have these types of multi-layered, multimodal networks is a problem which has received a lot less attention than the classic problem, which is a single layer. So that's the topic of the talk. This is sort of the ultimate multi-layered network, which is the network that allows us to live and thrive, where you have layers from the molecular or even perhaps the atomic layer, which isn't shown here, that rise up through this multi-resolution, multi-scale network. All the relations here are different at each layer. We're out to, from the molecular to the social network of interaction, one has this incredible diversity, right, of the way that information is exchanged within the layer and between layers. So we're going to address a much simpler problem than this in this talk, but I think this is the ultimate goal, right, in integrated omics of human life. So this is a more specific goal, which is the 5D nucleome that Indica has been a pioneer in exploring, where there's 5 layers. There's the layer that corresponds to spatial organization of the chromosome territories within the nucleus. And this spatial organization is then coupled with the functional layer, which is the gene expression layer. So transcription, translation into ribosomal proteins or other types of methylation or what have you. And so time is a final layer. So that's the 5 dimensions in this problem. So this just shows over time, this might be over the cell cycle, where these chromosomal regions or territories change. They configure in such a way that promoters and enhancers along the DNA strands for each chromosome come into contact with each other and then produce a expression, right, of messenger RNA that then causes a metabolic conclusion of some sort. So the 5D nucleome I'm going to come back to at the end. But this is the basic issue with the network observation models for multi-layer networks that we're going to be talking about. There are 2 types of network data. One is where the edges in the network are relations between vertex pairs. So, and the other one is where edges are observed similarities, like inner products between patterns of action or reaction of the agents themselves. So this comes out of a paper that we published in a different area, non-genomic area. These are recommender systems, social collaborative information retrieval, where people listen to a given artist in a particular genre of music. The users of this last.fm, where the data came from, which was an old site where you could download different types of musical programs. Each user has their own preference patterns, which is described by a matrix of genre and artists. And so one can look at similarities between this matrix across the users to infer or to cluster users into different groups and do what's called matrix completion to fill in a 1 or a 0, which indicates whether there's a preference for this genre and this artist or not. If the user doesn't have an entry, hasn't listened to that artist or that genre before, but has listened to other artists and genres that are similar to other users who have listened to that particular artist. And then coupled with that, there's a social network, friendship network, that describes declared friendships, declared relations between users of this network. So these are two different types of networks. There's the network that is formed by similarity between the preference patterns of users, and then there's the networks that declared friendship, declared relationships. So we have a similar type of network here, right, with the technologies that are used to explore the 5D nucleome, the Hi-C, which forms a matrix of connections which are basically contacts. And this is called the contact matrix or contact map across a particular chromosome or across different chromosomes, which each contact is a particular relation which is confirmed by the assay to apply a spatial proximity of two genes or two regions along a DNA strand or two chromosomes. So this is our observed relational data, and then we have gene expression data, which is over gene index. Same index is indexing these columns. If you divide these into gene territories or gene domains, and we have multiple measurements of these genes. So these are the two types of models, and we're going to have to deal with them a little bit differently. At the end of the talk, I'm going to give an example where we have basically a Poisson type of model for the observations from this Hi-C, and a Bernoulli kind of model for a thresholded correlation matrix that describes the similarity between different gene expressions. So one of the things that's important to pin down are what are our objectives, right? So in a network, such as this network here, one can identify nodes as being positioned in the network in different ways in the sense of interacting with their neighbors. So if these are people in a friendship network, for example, you have people that have high centralities or low centralities defined relative to different criteria. So between the centrality is a centrality measure which is defined as the number of shortest paths between all pairs in the network that pass through that particular node. And the high between the centrality basically characterizes the importance of that node to the connectivity of the network. Because if you remove that node, you remove a lot of the paths, those shortest paths that would normally go through the node. They become longer. In fact, some of them become broken, perhaps, so you don't have a path. Eigenvector centrality is being connected to highly connected neighbors. And closeness centrality or degree centrality is having degree centrality is having a high number of neighbors. So finding vertices of high centrality is important. Detecting clusters of vertices like this one here is important. That's the community detection problem. And then performing statistical inference so that we can take this, say, over time, or take two such networks under different treatments and test hypotheses about topology and about relationships that exist, given that we have, say, these Poisson distributed edges, weights that are counts, integers that are random, right? So you have to account for that randomness. Centrality has been used a lot in biology. I just show a couple of examples here. Virus strain genotyping, where a H3N2 flu virus, influenza A, gets the viral sequencing is performed on each confirmed case across the world, and then you look at distances between the sequenced string to basically trace back the lineage of the virus. And so centrality plays a big role in describing where those sort of critical passages of the mutations of the virus as it is passed along, communicated from one person to the next. Network disruption is another example where in cancer, for example, this paper claims that the degree entropy, that is the variation of the entropy across the entire network of the degree of the nodes, is a good predictor of resistance to treatment. And so that's very, very key. This is a picture from a manuscript that's in review that Indika and I are co-authors on, which shows network centrality of various types, eigenvector degree centrality between this centrality, which are applied to the high C network, this network here, where the edges are extracted by basically thresholding this network and forming a binary adjacency matrix that describes the existence of edges between pairs, row column pairs of that correspond to genes in this case. And you can look at the centrality measures as a marker for each one of the genes as a proxy for some attribute of expression, gene expression, through RNA-seq. And so in this paper, we hold the position that between the centrality, degree centrality, eigenvector centrality play very different roles in linking form to function, right? And this is another example from another paper that appeared just recently on using what's called multiplex centrality over a time series of snapshots. In this case, it's the cell cycle using Hi-C from an experiment that Indika performed in his laboratory 4 or 5 years ago, and then being able to relate using a clustering through this multiplex degree centrality, which measures basically on the one axis the overlapping degree, that is the degree to which the centrality is persistent over all time in the cell cycle, versus the multiplex participation coefficient, which is the opposite, how concentrated that centrality is. So it doesn't change over the cell cycle. And so what you can see is that you get these clusters of transcriptional expression coming from clustering the Hi-C data that yield these two clusters here, which correspond to sort of an early cell cycle during the duplication phase in the transcriptional before transcription, then the duplication of DNA, and then followed by mitosis. So these, again, can be used to sort of relate, this multiplex decrease centrality can be used to relate high C to expression. We haven't yet actually combined two dependency networks, which is what I'm going to aim to do at the end. So community detection is a problem which is very simply stated as if you're given some network that you think there might be a pair of communities or more than two communities, in this case there's a pair, then the objective is to basically reorder the rows and columns of the adjacency matrix, which describe the edges, right, between these nodes, so that you render this almost block diagonal, right? So that the off diagonals, which correspond to the C here, corresponds to the edges that connect the two communities together, is dominated by the block diagonals, which have much higher connectivity within each one of the clusters. So there are various ways of doing community detection. There's combinatorial methods, such as min-cut, which is a highly complex problem. There are parametric methods based on stochastic block models and other approaches. And then there are data-driven methods that use spectral clustering, spectral graph harmonic analysis to find this cut, to basically find the reordering of nodes into community 1 with N1 nodes and community 2 with N2. So the principal question is how robust are these community detection methods to noise? What do I mean by noise? Well, let's say there's some spurious interactions that are either measurement errors, or they may be interactions that actually exist in the mechanism, but that are minor interactions relative to the interactions within the communities. So finding the number of communities is another important problem. Here there are two, there could be more. And then prediction of the accuracy with which you can detect that community is another one, so that you can post hoc come up with, after you've gotten your community detection algorithm has given you a result of this partitioning is the most likely community organization in your network. How reliable is that statement, given that there could be random edges or spurious edges that are adding noise, right, to this network? And so this is the noisy community problem, the detecting communities with noisy edges, where one has a ground truth model which has very few edges, maybe even zero edges between the communities, but the observed model has all these noisy spurious components. And so the adjacency matrix can be basically looked at as an additive noise model, where you have the ground truth A0 adjacency matrix, which has this perfect block diagonal structure separating these communities completely, plus this noise term, which are these off diagonal Cij terms that correspond to these cross couplings between communities. So the objective using this type of method is to figure out what is the effect of these noisy edges on being able to detect the ground truth, given that the actual observed graph has this adjacency matrix here. So algebraic connectivity is one method that's very widely used in spectral graph clustering. And basically, if you have two communities like this, in the red and the blue community, that have very few edges connecting them relative to the number of edges connecting the communities here. You form the adjacency matrix, which might look like this. The zeros are basically deleted from this picture. And then you take something called the graph Laplacian, which basically subtracts the degrees of each node from the diagonal. And then that graph Laplacian then takes this form here, and then you form an eigendecomposition. And that eigendecomposition turns out that the first eigenvector is constant. The first eigenvalue is 0, because of the fact that you've basically summed over the columns to get this degree matrix here. And the second eigenvector will almost perfectly distinguish between the communities by simply ordering that into the negative and the positive signs, reordering, that is, the order of the nodes. You'll get one community that has negative signs and the other one community that has positive signs in the second eigenvector, also called the Fiedler vector, due to Fiedler, who developed this method. And so this is proven only relatively recently that this type of technique will give you an equivalent community detection as the min-cut algorithm, even though this is a much simpler order p cubed is what the eigendecomposition requires, whereas the min-cut is an exponential complexity. That, of course, is only the case when the communities are well separated. There are lots of other related approaches, like the modularity method, which I'll talk about in a little bit in passing, that does a similar type of eigendecomposition, but directly on A. So all these methods are sensitive to spurious edges, and so one of the ways the spurious edges can come in if these are correlation networks, so you've taken an RNA-seq assay, you compute the correlation matrix over all of your replications or all your time points, and you then threshold that to get a matrix, a matrix with adjacency matrix that can then be rendered as a graph. Then that graph is going to have some false edges and perhaps some false connectivity. So hubs of various degree that I've shown here. And so the characterization as a function of the threshold that's applied to the sample correlation matrix to obtain this graph as a function of the number of nodes in the graph, the number of genes in the genome or in a particular chromosome, and as a function of the number of IID sample replicates, the number of time instances that you use in order to construct the sample correlation matrix. We want to be able to discover what is the nature of the false positive edges or hubs that one might encounter. And so there's a whole theory of this that we developed a few years ago. It's some very late breaking generalizations of that theory, which state that there is a phase transition that depends upon the threshold that you use to extract your edges from your sample correlation. And it also depends upon the number of samples you have to construct the correlation and the number of variables. In a nutshell, if you have too few samples for the large number of variables, then you're going to get a lot of false positives. This is actually this is an empirical result for a network that was constructed based on ground truth that had no edges, right? So we had basically independent variables, independent gene expression values over the replicates. And so this is a histogram of the number of false positives, and so for almost any correlation threshold that you apply, you're going to be picking up false positives if you only have 10 samples or 100 variables. And so what we're able to do is to derive a mathematical equation that describes exactly where this threshold occurs as a function of n and p, and this threshold is on the number of false positive detections of nodes of degree greater than or equal to delta, right? So if delta is equal to 1, then you're looking at
look for these kinds of findings. And the examples are here. So here are the 13 highest rated by molecular function, and I've started a couple. This first one and the fourth one both have extremely high scores for transferase activity. Well, you could say, that's not very specific. But in fact, it's for sphingosine and acyl transferase. This is a very important function in the lipids in the brain. And it's true for this one down here also. So this would be a terrific target for experimental investigation. And then here's another one. This is a cytoskeletal binding protein, we say, and we think it binds spectrin, which is important. And then if you look at this in three-dimensional structure, it actually predicts a structure which matches very closely an E. coli protein for sugar transmembrane transporter. So this gives a huge boost to looking rationally for functions of these proteins. So the summation of all this is the metrics. We made a lot of progress. We're now at 89% of total predicted proteins and 15,798 canonical in the peptide atlas. There's still 2,186 missing proteins. For our chromosome 17, we are very close to the next 50 missing proteins to be found. We've already got 43, plus the excellent mass spectrometry stranded peptide I showed you, plus 35 high priority PPI or MS targets, specifically identified. And finally, we have at least 13, maybe up to 33, molecular function and biological process, uncharacterized PE1 chromosome 17 proteins with now high confidence functional annotations using our predictive methods. So I hope I've given you a sense of the broad picture of DNA, RNA, and proteins, the essential role of proteins in going from genomes to cells, and enough examples of computation and the underlying mathematics, which is embedded in all of that, to make a contribution to the theme of this workshop. Thank you very much.
Any questions? And also,
Is there a way to take all this research and, say, increase the human lifespan?
Well, that's a good big question. I'm a member of the National Academy of Medicine, National Academy of Sciences. We have just launched a initiative with all the new excitement in molecular biology and many things, not molecular at all, how to be healthier from an early age, from before you're born, to have healthy features, exercise and good diet and avoid nasty exposures and try to avoid the disease that cut lives short quite dramatically. But the goal is not just to live forever, because the quality of life is so important. So our goal is to enhance the healthy lifespan of humans. And there's going to be a competition, big prizes. People come up with ideas and to get some moderate funding for promising experimental approaches, and eventually there are a big number of prizes we anticipate for people who actually show they can make a big difference in some feature or other related to healthy aging. There's a lot of interest, and donors are interested in this. It's not a big surprise. And it's a little like the X Prize, for private sector space flight. So we'll see how that plays out. In the meantime, we have pressing problems dealing with diseases that are taking a horrible toll late in life, like Alzheimer's disease, neurodegenerative disease that impair cognitive functioning, thinking, making decisions, being aware of your emotions and your intellectual challenges. In the United States and all around the world now, this is an epidemic of degenerative brain disease, and figuring out ways to, fresh ways, to understand the disease process, to recognize almost surely that there are many different causes of it, like what look like similar patient problems, and to find ways to intervene either earlier in the process or moderate the progression once it's started. All of this is a big subject in the United States and all around the world. And in principle, this kind of basic research will play into all of that, of course. There's a new technique called gene editing, CRISPR-Cas9, previously zinc fingers and talons. There's a succession of these molecular methods enabling to change certain genes, maybe to correct a mistake in a gene, a mutation in a gene. This is all promising, but early stage. Yes?
Yes. Hello, Professor. Because I haven't done any research in this kind of area, so maybe my questions are done this very we know that we have like a gene regulation network, which maybe they're in progress, but we have that kind of network. So I'm wondering whether we have the protein interaction networks, and if we have, like how good are these? Can we utilize them to help us to like identify cell groups or to do like clustering or something?
Yeah, that's a wonderful question. Questions about networks. So biological functions, I appreciate you bringing this up, are organized in pathways and networks. So pathways, for example, if you look at metabolism, if you eat a sugar molecule like glucose, it goes through a metabolic pathway step by step. We know every detail. It was brilliant research of 2 generations ago. Still a lot to learn. If you ask about the pathways in the brain affecting thinking and Alzheimer's, we don't understand nearly enough about those pathways. Networks are a very commonly used way to explain interactions of proteins, so these protein-protein interactions I've been talking about, protein-lipid interactions, protein-nucleic acid interactions, and you can ask how well connected are the activities of 2 different proteins, or the isoforms, like the splice isoforms, of the same protein or of different proteins. So in fact, one of our postdocs at Michigan from South China published a whole series of papers with me on this, on the genome-wide, proteome-wide prediction of networks at the isoform level. At the gene level, I think is a little unimprecise, because we know that the gene is a mixture of these different transcripts produced by splicing and their proteins. So we think this is a major advance, but it needs to be validated experimentally. And there's been a lot of interest about this. Yes, indeed. If you know the term systems biology, networks and pathways are the heart of systems biology. Good question.
Okay, I think, Michael.
Thank you. Thank you for your talk. How is the human protein project finding and characterizing, helping and affecting the drug discovery, new drug discovery? New drug.
For new drugs, yeah. I think the fairest answer is to say we are not well connected with new drug discovery. However, we are providing the substrate for the biotech and pharmaceutical companies to have much more information for new drug discovery. The biomarker work is at the heart of what the drug companies need. They need to have clear evidence of what the target should be for a drug that could be designed or could be used from previous work to modify breast cancer, modify Alzheimer's, modify blood pressure, and this is a vast enterprise. There are several proteomic scientists at big companies like Roche, GSK, and others that have participated in the HPP. So they bring their own interests, and we always welcome them. We're very eager to have company participation. But usually they're reluctant to share their findings. So, we'll work with anybody, but the goal of this is open source, open sharing, maximal progression of the field, and we're thrilled if anybody can turn it into benefit for patients. We don't have good examples yet.
I think we have tea outside. There are a lot of good questions, and Professor Oman will be here till next Tuesday. And then please make appointment with him, and you can discuss more good questions.
Actually, only until Monday morning. Early Monday morning. So you have to catch me by Friday.
So you know his email number, you can write to him and make appointment. And we are highly encourage that. And that's why this is month long, and they come and stay for a week.
Well, I'll stay around the rest of the afternoon. Anybody, while it's all fresh, is a good time to think about your questions. I'm happy to have emails or follow up. I'll be around tomorrow afternoon, Thursday afternoon. I don't think we have anything here on Friday, do we?
No. So you guys have a lot of time in the morning.
Catch me when you would like. But
Method that we developed for community detection with adaptive model order selection. So it turns out, I didn't tell you what these are, but that you can sometimes actually write down the phase transition exactly for particular types of spectral clustering. For example, for modularity, Newman's modularity method, it turns out with two different groups that in a stochastic block model, that this theta star is equal to the harmonic mean of the probability theta 1 times theta 2, or the harmonic mean of theta 1 and theta 2, which are probabilities of community detection, community connection. In other cases, you can find lower and upper bounds, and you can use these to basically adaptively select the right number of clusters. And I'm not going to discuss how this works, but this will appear in a paper that's in press that allows you to start from network data and basically completely automate the process of both selecting the number of clusters with a statistical guarantee on the confidence in those clusters, and then identify your communities. So there's centrality in community detection in multi-layer networks is a different story. We've talked about single layer so far. I'm going to talk about a multiplex network where the only connection between layers is within an individual node. There are no cross-node connections between two layers. So there's methods for these multiplex graph analysis that have been developed. Lots of work in this area. I'm not going to describe them. I'm going to talk about a particular method before going on to the relevance network latent variable method that will combine multimodality network layers. First starting with a very simple idea. And the idea really is, you don't have to look at the slide, the idea is you have multiple layers of a network. What if we simply take a weighted combination? Flatten the network into one network, and then perform community detection on that flattened network. And if, depending on the weights that you use on each one of the weighted combination layer averaging, you can get a different degree of community detection accuracy. So you look at a convex combination, flatten the multi-layer network into one layer using a set of weights, and then you can apply this adaptive model order selection method I just mentioned, or any other method for spectral clustering. So we can do a phase transitional analysis for this case very simply, and we can also find an automated weight selection procedure. The problem, of course, is that you're scalarizing the problem, right? You're taking a rich multi-layered structure and you're compressing it into a single layer. That's likely not to be a universally good thing to do, even if you get to choose all of these weights, because at best you can reconstruct a convex combination of your multi-layer, but you can't construct, Pareto frontiers or other non-convex bodies that might be relevant. This just shows the phase transition analysis. I'm not going to mention that. This is a picture of the phase transition analysis and how the phase transition analysis accurately predicts, even in a finite number of samples, the actual behavior of a multi-layer convexification. I'm not going to discuss that. There are some experimental results where we use this on the HIV genetic interaction network that Domenico analyzed, comparing it to a lot of other algorithms, with showing that this method that we proposed, this multiplex multiple order selection algorithm, gives you a much higher conductance and normal cuts than the other algorithms. This is with uniform weights. Mimosa has optimal weights. They're chosen in order to give you the best possible phase transition. I'm not going to talk about the Pierre-Auger co-authorship. There's another experimental result. We had ground truth, which is the party affiliation of the 109th Congress, congressmen of the 109th Congress, were based on their voting records on various bills on the budget, on energy, on security, to see if you could reconstruct the communities, that is, the Democrats versus the Republicans in the U.S. Congress. And we show again that using this method, you can do better than competing methods, except for the bills that involve security, because that's when Republicans and Democrats tend to vote the same way, right? So you wouldn't be able to separate them anyway. So I'm going to finish on this multimodal relevance network, and this is where we have interlayer dependencies that the other methods don't model directly. So we're going to use a latent variable model, which is nothing more than a relevance network or a pair of relevance networks which are joined by a latent variable z. So you have two networks, say x and y, and what we're going to do is, we'll just look at this bottom picture here, we're going to say that x and y condition on this latent variable z, which accounts for all of the commonalities between the two layers, the dependencies between those two layers. If you're given that latent variable z, then x and y are conditionally independent. So that's all this slide says. And this just tells you how to measure independence and characterize independence, for example, for the Gaussian case, where this could be described completely by the partial correlation structure of the covariance matrix. So this is just a picture of this latent variable multi-layer relevance model, where you have two adjacency matrices that govern these observed matrices, and there's a latent variable y that controls the dependencies between the observed matrices. And so you have conditional independence of W1 and W2, these observed matrices, given the latent variables. And this then we're going to apply to the 5D nucleome. So this just shows you the experiment. We're going to take this experiment from the 2015 paper, PNAS publication, that shows just the high C samples and the RNA-seq samples at successive time points. We're going to look at the eight time points from 0 hours to 56 hours, and those are going to be then, for each time point, going to be characterized by the high C matrix, of which I only show the marginals here. The high C matrix, of course, is an enormous matrix with all the cross couplings between the domains on different chromosomal strings. These only show within chromosomal contacts. Likewise, we have a functional expression layer. So this is the chromatin gene structure layer. the functional expression layer. This is the correlation matrix of gene expression. This is just for chromosome 4, because I didn't want to show the entire genome here. And this is the partial correlation matrix that I didn't explain, and I'm not going to explain any further because I don't have the time. But that the thing I want you to note here is that this is a much sparser matrix, and therefore we're going to use it in terms of extracting a network by thresholding the partial correlation as opposed to the correlation matrix. And indeed, the partial correlation, as I told you before, is what carries this Markovian property of Gaussian, of Gauss-Markov graph processes that makes this the naturally adapted matrix to threshold to construct that functional network. So we have this for chromosome 4. We have this this chromatin gene structure network of contacts between genes along the chromosome, and then we have this gene-to-gene expression similarity measured through the partial correlations. And so you can use the correlation screening theory that I mentioned before to threshold this. We have 24 samples, or 3 replicates, 8 time points. On the chromosome 4, there were 764 non-zero expressed genes. So the critical phase transition threshold, if you go back to that plot that I showed, you can pick off the critical phase transition is at 0.72. And so that's great if the biological significance of similarity between expression at a correlation of correlation similarity 0.72 is reasonable. And then a 5% false positive threshold is a slightly larger threshold that ensures that only 5% at most of the edges will be false under a sparse assumption on the correlation, the true connectivity. And so then we have these 2 networks, the RNA-seq correlation graph that's been thresholded according to this phase transition analysis, the high-seq contact graph. This is Bernoulli because we thresholded it to 0, 1. An edge exists or not. The correlation is above or below a threshold. When I say correlation here, I'm referring to partial correlation. And the high...
a very different expression profile, then when they differentiate, that should also be different. So within the tissue environment, you will have input by the tissue, free fatty acids or whatever the adipocyte signaling is that's different in obesity as well. Mm-hmm.
So Lindsay, this is a very nice set of studies and a huge problem. In fact, in America, it's been documented for the last 40 years. Percentage of adults, the percentage of adolescents, children, over the barrier, the threshold for obese or overweight has been growing rapidly, and there's been no amelioration by people. One of the problems in biomedical research is that it's very useful to study animal models, mouse models, to explore possible mechanisms. But there's always the question whether the mouse model has anything informative for the human. That's particularly a problem when the mouse model may not even predict for other mice. So educate us, please, about where you think the models you're studying are most likely to be informative and transferable, and where you've discovered that they may not be.
Right, so I think where they're useful is in terms of understanding systemic response, so whole body response, circulating cells, these things, and as well as understanding the lineages of cells that are involved. So things like lineage tracing, which we can't do well in humans yet, is where that will be very useful. What was yours? You had a second question?
Parallel to bariatric surgery in the mouse?
There is, yeah.
Only 50% of them respond?
I don't know that. Yeah, I don't know that. Certainly at the tissue level and the cell level, the markers that identify immune cells have been found to be very different. So I think these kinds of studies where we actually have cohorts of human patients are extremely important. And we weren't sure when we started, there were very few papers published on what markers were there, what to use, and so we struggled a bit in the beginning to find a good set of markers. And we still don't know. I think there's a lot to be done here. But it certainly wasn't the same as the mouse cells. So.
Yeah, I think Dr. Oman, you touching the, I think more essence here is that whether the mouse models, how that reflects human systems. I always had this question myself too.
Yeah.
And maybe some sort of general functions like circadian maybe reflect in animal or human, there are similarities. But that's a big question whether the like obesity or cancer, I don't know how that reflects in animal model versus human. That's where maybe the reprogramming might help.
Next question. Yes. You mentioned reprogramming in the model and in the humans, as well as your own interest in reprogramming for therapeutic trials. But understanding that reprogramming is part of the disease process should be important.
Absolutely, and I think this is where to understand between lean and obese, but also between obese, the people who actually get diabetes, and some obese people will never get diabetes or a metabolic problem. So to understand those differences in terms of their gene expression signatures, to understand what is different about those will be very important.
I think we have enough time. Any more questions?
I know this is not a biology audience, so if you have any basic questions too, I'm happy to answer.
You want?
Yeah, I have a very stupid question. You mentioned in the middle of the inflammation cells, when people lose weight, will not disappear, still there. So does that suggest the immune system have to be adapted to clean those cells? That's not so easily reprogrammable.
And I think, so this is where we
The question.
yeah.
Make sure everybody understands the question.
Oh, yeah, so how, can you restate what you said?
I just wonder how the cells with inflammation cannot be that maybe exists
To resolve.
Yeah, after people losing the weight.
Right, so.
How the immune system can.
Right. So I had mentioned that the immune cells within adipose didn't go away after weight loss. So this is something we don't know much about yet, because we don't know sort of the timing of when this might resolve. And certainly there's a component of tissue damage where we see, and even in the human we did some studies on fibrosis. So the matrix that holds all the cells, right? So that changes dramatically as well. And so all of these things, I think, contribute to these cells staying within the tissue with weight loss. And there's a lot of tissue repair to happen to resolve and to reconstruct what the tissue was before. But we don't know whether it will ever go back. And this may be one reason why with obesity you have, for all of us, it's difficult. So once you reach a state of obesity, it may be very difficult to go back. So there may be some permanent changes that happen if you go long enough and have a chronic obesity situation. Maybe 1 to 2 years, okay, you can lose weight and resolve completely. But longer than that, we don't know the time scales. Certainly, but I think the immune system definitely has a function in repairing the tissue and resolving the tissue damage that can happen.
That would make childhood obesity all the more damaging.
Absolutely. So consider when this happens in adulthood, we see this, but then in childhood obesity, the long-term effects can be pretty serious.
a question about genetic inheritance of risk for obesity, and specifically what that suggests about educating parents who may be obese or maybe were obese as children about what they should be doing about their own children in a more urgent sense. If you're talking about priority populations for intervention, I'd be interested in your thoughts.
Yeah, and certainly genetics should play a role. It's difficult, obviously, to separate the genetics from, the cultural and family culture behind eating. But I think that will be a huge key to modifying behaviors. Is number one, education and targeting the parents who need it the most, and it tends to be associated with socioeconomic status and education level. But I think certainly for all of us, as we know, food is good, and we love food, and we love to eat many times and eat food that tastes good. And so I think it's going to be many factors that we have to address. But I think certainly targeting families and parents, and in particular, will be very important.
Are there genetic differences between those adults who have the diabetes problem in addition as opposed to those not?
That I don't know. Yeah. So I think we only looked at, in my data, looked at gene expression. So not sort of the genetic backbone of what variation was there, but certainly that could play a role. Mm-hmm.
So I really like the Yuan's question. So that means there's some sort of memory, right? Like a cellular memory, how that is affected by obesity. Is there evidence for?
Yeah, and I think that certainly can be playing a role. Yeah, so I think that because these cells are so tuned to their environment and sensing their environment, they are programmed by that to behave differently. So when we're talking about monocytes, they circulate in the body for a few days or a week, but then they're replaced by new monocytes. Going back to the origin of those monocytes, in mice we see that the bone marrow actually changes as well in obesity. So when they're exposed to obesity, the cells in the bone marrow become the blood cells, and those cells then are different from how they were when they were not exposed to obesity. So I think in that sense it should happen from the progenitor all the way to the differentiated cell, and some of those cells stick around for longer than others. So when we consider an adipose tissue macrophage, those are relatively long-lived compared to monocytes. So if they infiltrate a tissue and differentiate in the obesity environment, those could stay in the tissue for much longer and promote inflammation and dysfunction within the tissue for a longer time. Stephen?
Yeah, as far as immune response, damage to cells, etc., do we see, is there a sliding scale between being overweight, obese, morbidly obese, or is there kind of a threshold where you start to see these kind of gene expression differences and other problems arise?
I'd say that's really unknown in humans.
If you have enough measurements, you can, enough samples, you could reduce the threshold so that you get a denser graph. On the other hand, what you're saying is correct, that there will be spurious discoveries in these shared communities. And what we're hoping is that we can develop a phase transition analysis that will apply equally to this latent shared community scenario as to the single layer case. So for the single layer case, all the theory is clear, right?
Yeah.
You can look at phase transitions, which will tell you about these probability of spurious discoveries, if you use this convex combination principle that I was telling you about. But on the other hand, that doesn't account for the differences in the distributions between RNA-seq and Hi-C that also would be important in determining these spurious discovery probabilities. But you're asking a question that we're actively working on and that we hope to be able to answer in the next few months.
Thank you.
Thank you.
And I have another question. All the graph we talk about is undirected graph.
Yes.
And I think, for example, if take gene expression graph, for example, there is several kinds of causality because some gene will just promote the expression level of some gene. So I think is it possible to take the time series data to make the graph or directed graph?
Yeah, great question. You know, in my hour-and-a-half version of this talk, I actually had a whole section on the directed graph story using directed information and time series causal models. So the answer is yes. The problem is, from a measurements point of view, is that if you want to extract directed relations, especially if they're of time series nature, so you're looking at a dynamical system, and you want to figure out what are the causal connections, what's the structure of the F matrix, right, in the dynamical system, you need a exponentially larger amount of data. So the amount of data required as you increase the number of parameters, and the phase transition analysis that I was showing contains this, the statement that I'm about to make, is that the scaling is terrible. The slope of the degradation of performance is very punishing. So you need a tremendous amount of data if you want to come up with directed graphs for large networks. So it's an open problem. If you're willing to make some shortcuts and some assumptions on the nature of the interactions, for example, if you might want to assume that they're just Markovian, so you don't have to consider the entire past. You might also want to think that they're only pairwise interactions that are important. You don't have to consider higher order tensor types of reactions. You can simplify things, but we still have this issue that at least today it's too expensive to do sequencing in order to come up with the sufficient amount of data to do causal analysis. That's my own opinion. But
it's a good. Professor Hiro will be here, and you can make appointment as I mentioned before.
Yeah, sure.
And room 6302 and I think 6302. Please.
Great. Yeah, welcome.
And you too.
Welcome anybody who wants to come in and talk. I'll be here till Friday.
Yes. Yeah.
Take advantage of this great set of talks that are coming up also, and I'm very honored to be participating in this series.
So
thank you.
Sony